So thank you, Yoni, for the invitation and the reception from everyone here in Israel. It has been a very good time these days here. So I'll try to put some more context in which kinds of data we have been working the last days here. And so it has to do with some stuff that Michelle was saying, Alan, and it will link to the next one from Sebastian. So, and at some points here I may skip or go quickly into some slides, but you can all have the entire works and the entire papers later on, if you like, from the website of my lab that I'll put in the end too. So as Moshe was saying, I just to know geographically, I'm coming from this state here in South Brazil. So it's kind of the end of tropical reef fish in the coast of Brazil. So the idea here is just to show that we can have different data sets that came from different perspectives and that falls into different disciplines like evolution, biogeography, macroecology, and in between we can have functional biogeography or something like that. And when we combine different kinds of data sets, we can go and explore different metrics like richness. In the case of different traits, we can go for diet, mobility, and then we can go for abundance and biomass and even interactions of species. So I'll give some examples. And of course we are studying reef fish, and we think reef fish is interesting because, and Michelle told a lot of things about fish already, reef fish. And it's a group that it's somehow feasible and easy to collect data underwater for populations and for a number of species, and then we can have size classes that we can convert to biomass as the main studies of Alan. And we can go for and collect data in a complex system that I think it's we have easier access than forests, for example, to collect data from animals in forests. And we have it's relatively easy to identify species in the field. Reef fish are usually very colorful and have different shapes and marks, and we can assign them for functional groups that we know a little bit more than different groups like insects, for instance, that are much more complicated. And we can sample almost the entire community of fish, usually using underwater visual censuses. So from taxonomic checklists alone, we can study biogeography, and in this case here we assembled a lot of checklists from all over the world, and then we analyzed the distribution. We could analyze the distribution of more than 6,000 species, as Michelle was saying. We had a working group before that met in CESAB in France, that we were working on these kinds of questions, and then we here in Israel we continue on these ideas. So Michel already showed this kind of graph that we can do with checklists. So you're already seeing this. So I'm just skipping. And when we combine our checklist with the presence of absence of species, and then we combine it with traits, we can go for that global maps also of different traits, combinations, proportions. So we can check, for instance, the frequency of small species versus the large species. And there's differences between central and peripheral places, for instance. We can work on redundancy, but this is something that Sebastian will dedicate the next talk to work on this kind of combination. So this will be Sebastian's next talk. We also looked at the importance of sizes, and in sizes were very related to dispersal and to establishment of fish in distant locations. That's why Michel was telling you about the different proportions of larger fishes and smaller fishes in the peripheral areas. We can also combine distribution and abundance of species, and for instance the species abundance distribution that Sean Connolly, that colleague of ours that were working with us this week, that just published a paper on commonness and rarity using these kinds of approach. We can do this with biomass distribution also. That's something we are willing to do more within our group, understanding more about these places that are not fished versus the fished places, more with more human disturbances and so on. And another thing that I would like to show is that I have been working with interactions also. We can scale down to interactions of fish. So we can count, for instance, bite rates, agonistic encounters, the frequency of agonistic encounters, richness of interactions. We usually did that by counting it and diver taking notes of it. But now that it's kind of easier to do that with these small cameras filming things, I will present an example of how we are working with this. So we deploy a camera, and we establish an area that we will be filming for a few minutes. Usually, in our case, we establish it doing this kind of films for 15 minutes without the presence of the diver, and doing this especially distributing it in different depths and different parts of reef. So at this point we established the area we will be filming. and then we go away and then get the data. Another example would be something like this. Let's see. For example, sometimes you are just an interesting part of this kind of camera using. So sometimes we put that, and then we can later on count a lot of fish. It's hard to count in the field, but if you go and stop frame by frame, you can eventually count the number of bites and number of agonistic interactions between fish, for example.
With a bait or without a bait?
Without bait, just randomly putting it in the reef. And sometimes even sharks appear to take a look at the camera. It's funny how when we stay counting all this stuff and then suddenly appears something very different, a turtle and a shark. So it's very funny. But then we can have some, so we can have this counting these interactions, as Michel said. Some of them may be really important to the reef. In the case of the herbivores, for instance, that removes or at least disturbs algae and may help corals. And then these we can, for some extent, try to measure these interactions like bioerosion and other kinds of indicators. And so in the end, we can come up with this kind of graphs that we can try to relate temperature to this. In this case here, we call the mean feeding pressure is it's a combination of the bites that the fish promotes in that sample and its biomass taken from visual sensors in the same area. So we can calculate and extrapolate a little bit for the reef. So another kinds of combinations come from when you have phylogenies from the groups. So we can combine and work with turnover of lineages and phylogenetic beta diversity, for example. Then we can combine phylogenies with traits. We can take a look at even at the evolution and diversification of traits within using phylogenies. Then we can combine phylogenies, distribution, and traits, and try to understand a little bit more on speciation, diversification rates, for instance, from tropical to extratropical places. And we recently did some analysis on that and seen that speciation rates in the tropics are really more than in the extratropical reefs, and all these kinds of calculations. Then we can try to combine phylogeny, distribution, and abundances, we are trying to relate abundances and distribution to relationships between species. For example, here we have 2 butterfly fishes in Brazil and in the Caribbean, and this species endemic to the Caribbean, regional endemic to the Caribbean in each different local place. It seems to be more abundant than this widespread species that seem to be more abundant in the peripheral place in the Atlantic. And so we can ask these questions like if regional endemics are more abundant at the local scale compared to widespread species, or it's the contrary. Michelle already showed us some evidence that endemics may be more abundant, really more abundant in peripheral places. So there's all kinds of questions to be answered looking at phylogenies, abundances, and distributions. If species that are originated in some center of diversification, and as opposed to peripheral areas, and how this could be related to abundances in the local sites. Again, this example, but we are trying to have other examples of fish that may be regional endemics and maybe more abundant locally within its area of within its regional area. And we don't work with this in this group, in this particular group, but we know that when we have good fossil record for some groups, we can extend this to deeper time scales and work with functional traits of fossils and try to understand a little bit more how communities evolve through time. So that's about it, and I would like to show here again the link if you want to download most of this research I have shown. Thank you for